Adik TV KPM.
Okay, terima kasih. Okay, now candidates for section C. Okay, as what I've mentioned earlier, 3 questions. You have to choose 2 questions, right? And both questions, which is question number 18, 19, or 20, depending on which question you choose, will give you total of 15 plus 15, which is 30 marks. Okay, so now I highly advise you to spend 25 minutes per question. So overall, since you are going to answer 2 questions, that would be 50 minutes altogether. Okay, so now students, only decide the 2 essay questions that you can answer confidently, all right? And do not draw your diagrams to represent your answers unless in the question stated with the aid of a diagram. Meaning to say that question wants you to draw a diagram. That way, eventually you have to make some explanation or elaboration based on your diagram that you have drawn. Okay? So now, candidates, I'm sure you have seen or come across these 3 types of questions that usually come in the exam, which is describe, differentiate, and explain. How do they differ with each other? Now, for describe, it requires discussion that is fixed and not influenced or exploited by factors or conditions. Example, like SPM 2020, all right, the recent exam. Describe allopatric speciation. Okay, and with the aid of label diagrams, describe the Michelson install experiment of DNA replication. Okay, ataupun senang cerita, describe ini, jawapan untuk soalan yang berbunyi describe hanya copy paste dengan apa yang calon telah belajar saja. Okay, now when it comes to differentiate, it requires candidates to give concrete comparison between subjects or parameters in the question. Okay, so one of the example question is this: differentiate between missense and nonsense mutations. Okay. Then explain requires discussion on unfixed or exploited conditions. Okay. Stated in the question, answers will be very dependent on the situation given in the question. Contohnya, explain how disruptive selection can cause speciation. And usually candidates, you will only answer about disruptive selection, but you can't relate with the speciation. So for question that read, explain, you have to be very careful. Okay, your answers depends on the keywords of the questions. Okay, now tengok jawapan untuk soalan yang pertama, iaitu describe allopatric speciation. This is suggested answers by me. Okay, it may not represent the actual answers from the exam, but usually it is more or less. Okay, so now P1 means point number 1. Okay, so when you're describing allopatric speciation, you can start your essay with intraspecific speciation, where a population is separated into 2 dims via spatial barriers such as mountain range, oceans, or river. Now, students, when giving out examples, giving 1 example is enough, is sufficient in the essay question. You don't have to write all the way from mountain range to oceans and river. Okay, so for the purpose of teaching, so I just give you all the answers that I can think of. So bagi 1 pun cukup dah sebenarnya. Okay. Okay, untuk yang P3, both dims are isolated from each other, and this leads to reproductive isolation. And then selepas berlakunya reproductive isolation, gene flow between 2 dims will be interrupted, ataupun interbreeding between 2 dims is prevented. Nampak dia punya flow cerita dia. Okay, so each dim undergoes different selection pressure that leads to changes in genotype frequencies. Prolonged separation causes the dims to genetically isolate. Adaptable alleles are passed on to the offspring, and then the 2 subpopulations can reproduce to form fertile offspring, and this leads to speciation. So students, you can see the flow, how the allopatric speciation occurs, right? How it's being written in such a way that describing the allopatric speciation. Okay, now next question. With the aid of label diagram, describe the Michelson install experiment. Okay, so calon kena lukis gambar aja, because the question says with the aid of a diagram. Okay, dan selalunya kalau soalan yang macam ini ada markah diperuntukkan untuk gambar aja yang dilukis. Okay, so ini gambar aja yang calon kena lukis. Okay, so lukis test tube tu dengan betul dan labelkan test tube tu. Okay, ini ialah sebelum transfer 14N, okay, nitrogen 14. Dan ini one cell generation, dan ini pula two cell generations after the transfer to 14N. Okay, so gambar aja yang betul dilukis 1 markah, okay, dan label 1 markah. So 2 markah selalunya. Jika graf 3 markah peruntukan diberikan selalunya. Okay, now candidates, okay, berdasarkan pengalaman cikgu, selalunya calon bila DNA ini mereka lukis garis macam ni saja. Okay, garis macam ni. Tidak boleh. DNA, you know, is a alpha helix, so it must be drawn in such a way that represent the alpha helix. Lukis macam ini. Dan mesti label dia. Alright, ini yang hybrid, ini yang light DNA, dan ini yang hybrid pula dalam test tube itu. Okay, now contoh jawapan ialah E. coli, candidates, this must be underlined because under binomial naming. Okay, mesti dia underline. So E. coli was cultured many generations in a heavy N15 nitrogen medium, so that all the DNA strands would be heavy. I'm sure you are aware of this, right? Okay, next, the E. coli produced heavy DNA strands because the heavy N15 is incorporated into the E. coli DNA nitrogenous bases. Okay, candidates, again, please underline. Okay, this is the name of the genus, the name of the species. Sila gariskan semasa menulis ini. Okay. The next, density gradient centrifugation of the E. coli will reveal the heavy DNA strand is at the bottom of the centrifugation tube, as the heavy DNA strand is denser than the normal DNA strand. Okay. And then the E. coli was then transferred and cultured, okay, in a medium with normal N14 nitrogen, so that the newly synthesized DNA strands will be light. Okay, so it's a part of the suggested answer. And then we continue. DNA isolated from E. coli cells after 20 minutes and centrifuged in cesium chloride. Okay, students? To separate DNA into bands based on density. Okay, and then the DNA strand was found to contain hybrid DNA strands of both heavy and normal nitrogen. That will give you the 8 point. And then DNA isolated from the E. coli cells after 40 minutes and centrifuged in cesium chloride to separate DNA into bands based on density. That will give you 9 point. Okay. Then the DNA strand was found to contain half normal DNA strand and half hybrid DNA strand. Itu markah yang 10. Right. Now, without any further ado, let's continue with the differentiation type of questions. Now, here I just want to highlight you when you answer this type of question, make sure you use the word like while, whilst, whereas to show comparison between two parameters. Okay, so like this. Okay, and like this. Okay, now usually candidates, okay, if you write one paragraph, you talk all about the missense mutation, and then on the second paragraph, you write all about the nonsense mutation. That is not accepted. You're not making comparisons. When you are making comparisons, this is how you should write your answer, right? That shows you are making comparisons between two parameters. Okay, boleh faham maksud cikgu? Jangan tulis dalam satu perenggan semua pasal satu bahagian saja. Alright. Last questions. Explain how disruptive selection can cause speciation. So penjelasan calon mesti menunjukkan macam mana speciation berlaku akhirnya. Okay, so disruptive selection occurs when a selection pressure acts against the intermediate group of phenotype and eliminates the intermediate group. That's P1. Okay, and then disruptive selection favors both the extreme groups of phenotypes. I'm sure you are aware of this, candidates. And then disruptive selection occurs when environmental condition fluctuates between two opposite extreme values. Then eventually population is split into two subpopulations, and then interbreeding or gene flow. All right, candidates, this is very important. Between the two subpopulations is prevented. And then the subpopulations are reproductively isolated and have developed different allele frequencies. So over time, the two subpopulations are unable to interbreed and eventually become two species, which is speciation. Right, now candidates, if 5 marks are allocated, usually, okay, the total points that you can write or allocated for you to get points or to get marks are more than the 5. Usually 6, 7, maybe 8 or 9. So I mean, say you can write more. So I can say that when you answer section C, write whatever that you know. Right? So it might give you high chances on getting marks.
Didik TV KPM